Hello. My name is Frédéric Douhard. So I work in the GenFIS team in INRAE Toulouse. It's a team of geneticists, but I'm also doing modeling. So Laurence presented you some work on resource allocation. So I also work on this issue. And I also work on the parasite infections. So I will try to give you some insights on the resilience to parasite infections. So the aim of this session will be to provide you an overview of the process of building a mathematical model. And this mathematical model is a model of host-pathogen interaction. So we have two entities. And we'll see different things. The first is how we can design a model to address a biological question. The second will be to translate this conceptual model into a mathematical equation. The third thing will be to run model simulation. So you've already seen this with Laurence on Python. So this will be with R only. And the last thing will be to try to estimate the model parameters starting from experimental data. Here is our issue. It's a gastro-intestinal parasite infection. So as you may know, this is a big problem for sheep production. Basically, we have two stages, free living stages on the pasture, where the eggs hatch and develop. And then the sheep, they ingest the eggs, and then they get parasitized. And this is where you can observe a production loss and even the death of the sheep. And then the sheep also shed the eggs in the feces, and this start over a new cycle. So we have several problems with this infection. The first is that the population of parasites, they are evolving, and we have more and more resistance to the chemical products, the enthelmintics. So the solution that we had before is less and less a solution, and we need to find the alternatives. And the second issue is that these populations, they also evolve with climate change, and the areas where we observe parasite infections, they are also expanding. There are several species of gastrointestinal parasites. Some are specific to cattle, some to goats, some are generic, and you can locate them in different parts of the gastrointestinal tract. But we will focus on this one, which is Haemonchus contortus, which is probably one of the worst. And this parasite, the problem is that we don't have a good a specific biomarker of the host resistance to this species. So this means that if we don't have very good antibodies that we could take in the blood and to identify what are the resistant animals, it is not working very well for this kind of parasites. So we could simply take the parasites that are within the sheep. So this is the adult parasites, and you can notice the red color is because they take the blood of the sheep. Unfortunately, this measure is not very convenient because you need to kill the sheep, and it's a terminal measure, and it's very heavy to realize because you need to take the abomasum and then to count the worms within the abomasum, and it's very boring, believe me. So another measure is to take the feces and to count the number of eggs that are in the feces. So it's very simple because you only need some grams of feces to dilute them with salted water, and then to count it with a microscope and a specific, cell. It's called a McMaster cell. And basically, it's a grid, and you will count the number of eggs for each range of the grid. And this will reflect the total number of eggs that you can have for a particular sheep. So fecal egg count is very interesting because it's very sensitive, because with just one adult female in the sheep, you can have an excretion of up to 10,000 eggs per day. So it's Haemonchus contortus is very prolific. So it's very easy to identify the presence of Haemonchus for a sheep. And it's also very interesting because when you compare the variation between sheep, you can see that there is genetic variation. So maybe you have seen this yesterday, but the heritability is between 0.1 and 0.3, basically, depending on the species. We could select for host resistance if we use fecal egg count. And this is what people do, actually. And there are two main ways of doing selection. The first way is to do progeny testing. So you will select the rams based on the phenotypes of the offspring. So basically, you have your elite ram, and you will measure the fecal egg count of the progeny that are exposed to natural infection on different farms. But the problem with this is that you need a lot of animals per ram, about 30 animals. So it's to get a good accuracy of the index. And you do not have a very accurate measure of the fecal egg count because you don't know the number of parasites that have been ingested. You don't know which species has been ingested because you have other parasite species, as I showed you before. And you don't know what is the timing of the infection. So there is a lot of sources of heterogeneity, and this makes the fecal egg count very difficult to interpret. So another way is to use artificial infections. So with this way, you will measure fecal egg count directly on the elite ram. So what we do is that we prepare doses of parasites, specific species of parasites, so for instance, Haemonchus, and we know the number of parasites that we will give to the host. So we have a control measurement, and we know also the timing of the infection. And with this, we have an accurate measure of the fecal egg count. So consequently, we need fewer animals, and we can take advantage of the pyramidal organization of the selection, because we do this on the elite rams, and then we can measure the consequences directly on the progeny where the rams will be used. So it's an infection, and we are working with elite rams. So it's not very, how to say, recommended to heavily infect your elite rams because they won't produce and they risk to die. So we focus on the beginning of the infection, and after 5 weeks, we treat the animals so that the infection is not too heavy and has no big effect on the rams. And we also work with relative small doses of parasites. And also, we make sure that the species of parasites that we use is susceptible to anthelmintic drugs. So now we are confident that we can use this kind of protocol. Yes?
Just to know, what is the heritability of this trait?
So it's this data here. It's between 0.1 and 0.3, basically. So it's moderately heritable. And if you compare, you're right also to ask the question, because there is a difference between artificial infection and natural infection, and the heritability is higher for this because we control more the source of variations. So this is also an advantage of this way. And we have shown that it's effective to select RAM. Still, we have some questions. One question is a major question is the side effects of this kind of selection on the other traits. And basically, the other traits, it's production traits. So this is the genetic correlation between fecal egg count and body weight in lambs. And you can see that the genetic correlation, they range from negative values to positive values. So it's very heterogeneous. And on average, it's slightly negative, but it's not. It's very close to 0. So here we have we don't know very well what are the side effects of selecting for host resistance based on fecal egg count. Just to remind you the interpretation of this genetic correlation, if we have a genetic correlation that is negative, this means that we have a cost of infection. So the relation is negative. So it means that the more you are infected, the more you lose in terms of production. And on the other way, the cost of resistance would be a positive correlation. So basically, these situations. But to separate these two kinds of costs, one important thing is to have a repeated measurement to measure properly the residual correlation in your data set. And if we want to focus on the resistant costs, it's also important that we focus on the beginning of the infection, because it's only after several weeks that the infection costs predominate. So if we want to measure the resistant costs, it's better to have a repeated measurement and a short-term infectious challenge. And this is why we use the protocol of artificial infections to try to measure the cost of resistance. So in this experiment, we used 91 female lambs, and we used an infection of 5 weeks, so it's the same protocol that I've showed you before, with 10,000 larvae of Haemonchus contortus. And we have repeated measurements. So we measured first feed intake. So it was a concentrate that was provided ad libitum, and it was recorded automatically. We measured the body weight, and we also measured the composition of the body with the dorsal scanning. We measured the fecal egg count, and we measured, finally, the blood hematocrit. So the blood hematocrit, it's equal to packed cell volume. I don't know if you have ever heard of this. So I told you that this parasite is taking blood from the sheep. And when you have a lot of parasites, you have a lot of anemia. So when you put your blood sample in a centrifugation process, you observe that the red blood cells portion is lower in your sheep that have anemia. So blood hematocrit is also a good measure of infection with this kind of parasites. Okay, so this is the data, and now this is the model that we want to build. So we want a model that will take as input the feed intake. So this was recorded automatically with the machine. We also have another input that is the parasite ingested. We know the number of parasites that have been taken by the animal, and we know the timing. And as output, we want all the traits that I presented before. So body weight, back fat thickness. We won't focus on muscle thickness, just back fat thickness, fecal egg count, and the blood hematocrit. And this is what we want to build. So basically, it's an energy allocation model, but it's quite different from what Laurence presented you before. It's much simpler in terms of assumptions. So we'll use energy balance approach. And in this energy balance approach, you have different costs, and we will try to measure the cost of immunity. Because if you look at nutritional books, they give you a cost for maintenance, they give you a cost for growth, but generally they don't give you a cost for this kind of thing. So we'll try to estimate this. And as I told you before, we'll use R. And I propose that we open the script and that we process together with the PowerPoint here. So you can open everybody received the script that I sent. You can open the main script on R Studio. So this is the main script, and you can open this button here to see the outline of the different scripts that we will use. But basically, we will have 3 steps. The first step will be to do an energy balance model without parasites. The second step will be to model the parasite dynamic within the host. And the first step, we will put together the energy balance and the parasites. And we will try to estimate the energy cost of host resistance. So I come back to the presentation here. The first step is to build the energy balance model. So what we will do is that we will use feed intake data as input, and we will predict the body weight and the backpack thickness. And the feed intake input, we will use a function to smooth the data. And I think you have seen this morning with Nick. So I think now you are experts on this kind of method. And with this input, we will predict the body weight and the backpack thickness. So basically, we have the energy balance. That is the energy intake minus the energy that is needed for growth and the energy that is needed for maintenance. And so far, we will ignore the energy cost of parasite resistance. So the energy intake is this, as I told you. The energy for growth will be based on a model that is already exists, that was published in 1997 by Gary Emmons. And basically, the P is the protein mass, and the wool is the wool mass of the animal. So all of this is protein. And this is growth rate, if you take the sum of the two. And this is the energy cost of the growth rate. So here you have the quantity of energy per day. And well, this is just the equations behind, but basically you need to know that the growth rate of the protein is this kind of model. So it's called the Gompertz model. And a major, well, there are two main parameters that are important to know that characterize each individual. It's this parameter that is the individual growth rate, and this parameter that is the protein at maturity, so the final mass of protein that the animal will reach when it will be an adult. Well, the wool is just a proportion of the protein, the wool growth rate, so we won't go further into this. And the maintenance is basically, well, more or less proportional to the protein mass. So the key thing here is that the protein growth is the driving force in this model. And then when you calculate the energy balance, if it's positive, this means that you have some energy that you can allocate to build body reserves. And the L here stands for lipids, And with the lipids, we will predict the back fat thickness because it's the fat on your back. So it's basically lipid. So this will just be equal to the energy balance divided by the energy value that you need to build 1 kilogram of fat. The body weight will be the sum of the different compartments. So the body protein, the body lipid, the wool, the hash, water, and the gut fill. And basically, we need to predict these things. And these things here, I won't detail, but it's just proportional. It's just proportion of body protein. So it's just a coefficient. It's not very complicated. So we have our body weight, and we have our back fat thickness. And the back fat thickness, well, it looks complicated, but it's just based on an allometric equation that I found in a publication. And when you transform this equation, you get this one. So it's very empirical, this prediction. The wool? Okay. So I did not indicate, but we, at the end of the experiment, we shaved the sheep, and we weighed the quantity of wool per animal. So in this way, we could predict the quantity of wool that has been produced during the experiment. Yep. And there was a single measure per animal to predict the wool growth rate, but actually you will see it's very small quantity compared to the whole body, so it does not matter. So now we'll try to run this model on R. So now we open our main script here. So first, we need to upload 2 packages. The first is dissolve, is to run ordinary differential equation on R. And this one will be to estimate parameters. So we will use this one later on. So we load the packages. Then you need to indicate your path. So everybody did this, your own directory. And we'll start the energy balance model. So with this function, we import the other script. So we import the energy balance model, and we import the parameters. So just if you want to have a look on the script, the parameters files, it's just the list of parameters that we will use. And I gave you a description of each parameter name and the value of it, and etc. So that you can know what is going on here. And the model, it's the model is this. So it's a function that we will run with the ODE function of the ODE package. So basically, in this function, you need the time, the state of the animal, and the list of parameters. This is the 3 kinds of input that you need to run the ODE. And all these equations, well, they are exactly the equation that I presented before on the slides. It's just that they are written in R. So for instance, the D protein is the dP over dt. It's the Gompertz equation that we have seen with the beta parameter that is key, that is indicating the growth rate parameter. And the protein M is the protein at maturity. So, well, I think it's not important that we go into each equation, but you can check if you want with the slide. So now we will import our data because we have some data with the body weight, back fat thickness, fecal egg count, and food intake. And as I told you, we will use food intake as an input. So just first look at the food intake. This is the data here. And as you can see, it's very varying over time, even if we are under control condition. So there is important day-to-day variation, but we see a trend to increase, and this is because the lumps are growing. But to use it in the model, it will be more convenient to use a spline. So we will just fit a spline before. So to fit the spline, I use this function. I'm not an expert on splines, so maybe there are splines that are better, I don't know. But then if you want to look at the results, here, where is my plot here? Yeah, so you have a smooth variation of the food intake. So, well, it's not perfect for instance. I assume maybe this point should be removed, I don't know. Well, I just took it as it was. So this is our input. And just to remind you, this food is concentrate, 100% concentrate. And we know the dry matter content of this. And we also know the energy value. So we can calculate the energy input that goes into the energy balance model. We'll calculate another spline, but that will be useful to calculate the quantity of feces. So it's the average mass-specific feed intake. So it's the feed intake over the body mass. Well, just a detail. And then we will set our individual parameters, because in this model, there are parameters that are individual specific. So as I told you, there is the protein mass at maturity, and there is the growth rate. So these parameters, they were estimated before. So I won't detail the estimation procedure. Just use the value that I indicated. And we can, well, change the value of the different parameters. And then we go to the step 4, where we need to define a vector of initial condition. Because when you run an ODE model, you need a starting point, so your initial state. And with this initial state, you can run the model. So our initial state here will be the quantity of protein, the quantity of wool, and the quantity of lipid. And we will use the default value that we have indicated here. And then we need to specify a time of simulation. And this was 5 weeks, so 0 to 35 days. And we will use a time step of 0.1 day. OK. So now we are ready to run a simulation. Is it OK on your side too? Yep. OK. So to run a simulation, we will use the ODE function. So from the ODE package, we indicate the initial condition, the simulation time, the model we used, and the parameter we used. And we will store the results in this data frame, well, in this output, and we need to transform it in a data frame because it's a matrix by default. OK, so now we have run the model. It was quite fast. And we will plot the results. So first, the energy balance. So as you can see, it's very similar in shape compared to the feed intake, which is something that we can expect. And you can see it's positive throughout the infection period. So this means that our animal here will accumulate fat. And this animal was infected, but so far we have ignored the cost of parasite infection. We can observe the other effects. So, for instance, yeah. the body weight and the bad fat thickness. And for this outputs, we have the data. So we started from here, yeah, back fat thickness and body weight. And you can see that the fit is not very good, especially for body weight and well, also for body fat thickness. So maybe there are some parameters that we can try to estimate more accurately. And this is what we will try to do. But well, there are a number of parameters here. But one hypothesis that we could do is that the growth rate of the animal has been reduced during the infection because they were parasites, so they were maybe taking some energy, and maybe the growth rate has reduced. So, what is the effect of different growth rates on our outputs? So this might be a first thing to do before going to the estimation procedure. We will try to see what are the effects of different values of the growth rate. So this is what we call some kind of sensitivity analysis for just one parameter. So in this step, we actually define different values, and we will try to estimate the effect of these values on the outputs. So we take values from 50% to 150% of the value that we indicated for the protein growth rate. And we will test the different models. So this is just a function to repeat the procedure over the different iterations. And then we will use the matplots to see the outputs. Okay, so we run this. Okay. So this is the output. So remember, this is the value of the beta protein, so the protein growth rate. And what do you see? In terms of protein, we have increased. So this is what we expected. But yeah, can somebody could explain to me why the lipid has reduced? Sorry? I'm not allowed to. No, you're not allowed. Yeah. Somebody understand why the lipid has reduced here? When? Yeah. Yes. Actually, the food intake was the same for this animal, and we have increased the allocation to protein growth. So as a consequence, the allocation to lipid has decreased. So this is just a balance. And you see that with more protein, you have less fat. And these are the outputs that we try to predict. And lipid is basically related to body fat thickness. And body weight is the sum of everything. But here you can see that it's the main effect on body weight is body protein, because it's in the same sense. So we can see that if we play with this growth rate parameter, we can maybe have a better value of the fit. So this is what we will try to do. So in the step 1.8, we'll try to find a better value for our data set. And for this, we need a function that calculates the residuals. Because to estimate the parameter, we need some function that calculates the difference between the model prediction and the observations. So this is this function, the resid function. And then with this, there is an algorithm that we will use. This is the NLS.lm. And this is from the package minpack. And basically, this algorithm it's an algorithm that will look for different parameter values that will explore the parameter space and will try to find the the parameter value that minimize the sum of residuals. So it's basically what you can see with a linear model, but with different methods. And I won't go into the maths, but yeah, the idea is the same. So you first need to indicate which parameter you want to estimate. So in our case, it's the beta protein. And then we can calculate the residual for the default value that gives a very bad fit. So for instance, if you run this, you obtain the residual sum of square. And you can see this value here. It's well, about 9. And now we'll try to find a better value. So we use the NLS function. So yeah, it has run already. And now we can compare the new value of the residual sum of square compared to the previous value. And you can see that there was an improvement. Yeah. So in your opinion, well, what is the effect on the beta protein, in your opinion, without looking at the next step? Do you think there was a reduction or an increase? Sorry? Yep. It's an infection, so we can expect that the sheep has grown slower compared to a normal situation. So when you look at the values, this is the default value, and this is the new value. And you see that the decrease was quite large. So now we can try to figure out the effect on this new parameterization on the outputs. So we'll run again the model with the new value. Up. So this is the new outputs, out new. Yeah. Okay. So the default value and the new value, and you can see that with playing with this beta protein, we have a better fit, but still the body weight is over predicted, and the gain in terms of accuracy was more of in terms of body fat thickness, where the fit is much better now. But still, there is something that remains unexplained, and this is maybe because we did not take into account the parasites. So this is why, in the second step, we will try to model the parasites. Okay, so is there any question so far? Yeah, was it clear, the energy balance approach? So let's go back to the presentation. The next step here was to ask if there was a cost of immunity in this energy balance, so if this can improve the model fit. So second step, try to estimate the host response to infection. So now we forget the energy balance, and we'll try to model the parasite dynamic. So now our input is the number of parasites that have been ingested, and our outputs are the fecal egg count and the blood hematocrit. And the two things that we'll try to model is the parasite dynamics within the host and the immunity. And this is why we call a host-pathogen model interaction, because there is an interaction in the two ways. So I remind you that we focused only on the parasitic stage, and we forget the free living stage because we are in the context of artificial infection. So this is a representation of the parasites going through the host. So we first have the number of larvae that have been ingested, and then these larvae, they will change in terms of stage. They will establish in the abomasum. And then in the abomasum, after a few days, they will change. They will become adults. And some will be males, and some will be females. And the females are the most dangerous here. Because they grow the more compared to the males, and they will take a lot of energy to the sheep to lay eggs and to produce a lot. So this is why I told you this. And this is why we have the last compartment, the adult females that lay eggs. And as for the energy balance, we have differential equations that give the rate of growth for each compartment. So basically, you can forget the equation, but the equation resumes the inputs and the outputs of each box. So here we have an input that, it's the initial number of larvae, and you have two outputs, the mortality and the transition to the other stage. So this is why we have a minus mortality plus transition to the other state. And the larva established, there is an input and two outputs, the mortality and the transition to the two adults stage. So this is the input, and this is the two outputs, mortality and transition to the adult stage. And so on. So yeah. And finally, you see that you have your compartment of adult females with the inputs and outputs. Well, in this model, we need to relate it to the outputs that we observe. So because, as I told you, it's very heavy to count the number of parasites that are actually in the sheep, because you need to kill the animal. But we have the observation, the fecal egg count and the blood hematocrit. And basically, we will say that the variation in blood hematocrit is proportional to the number of parasites that are in the host. And each parasite stage will have an effect on blood hematocrit. So this is the relative effect of each stage, the omega parameters. And this parameter here will be the highest because it's the parameter of the adult females that are laying. So this is the most important impact on the host. And the fecal egg count is just proportional to this compartment, the number of adult females, times the fecundity rate. So I told you it can be up to 10,000 eggs per day and per females, and divided by the quantity of feces that the host is producing. And you can see that you have two kinds of parameters. You have parameters that are constant, that won't change from an animal to another. And you have parameters that will change. And these are basically this one and this one. And this is based on assumptions that tells that host resistance is controlled by at the level of the establishment, and it's also controlled at the level of the fecundity. And it's also controlled at the level of mortality, but we won't consider this option. So we'll only consider individual variation in terms of parasite establishments and in terms of parasites fecundity. So now we can jump to the model in R, and we'll try to run this one and try to see the effects of different parameter values for these two parameters. Okay? So now we are at step 2. So we need to import another model, which is the parasites model. Once again, we need to define a vector of initial conditions. So this is different from before because now our state variables are the number of larvae intake, the number of established larvae, and then the number of adult parasites. And we need also an initial value for the blood hematocrit of the host. And the initial value of the fecal egg count, it's 0, so we don't put it. Okay, and we state that the initial value of the larva intake is 10,000 because it's the doses that we gave to the animal. Yep. So now we can run this model. So it's the same thing that before for the energy balance model. You can find more details on it if you open the script parasite model. So we'll store the results in the same way as before. Out parasites. And now we'll try to look at the outputs. Tac, tac. Okay. So normally you have this figure. So here you have the number of larvae intake. So this is when the larvae they go from the mouth to the abomasum. And the green line is the number of larvae that establish within the abomasum. So you see that there is a lot of mortality at first, which is normal. And then only a small number of parasites succeed to establish. And then once they are in the abomasum, they remain in this place. And basically the number remains the same during a few days. And then they emerge to become adults. So you have one part that emerge as adult males, so the blue line. And the brown line is the number of adult females that are not laying, that are still immature. But progressively, this compartment decrease and this one increase. So this one is the number of larvae, number of parasites, females, that are laying eggs. So this normally should be proportional to the number of fecal egg count that you will find in the feces. Yep, is it clear?
So the sum of the non-laying and laying female are equal to the sum to male?
Sorry?
If you add the curve of the non-laying female plus the laying females,
yes,
it's equal to the curve of the male?
Yes, because, yes, exactly. Because the proportion was, the sex ratio was 50%. So, yep. But basically, when you sum all the compartments, which give you the worm burden, the total worm burden, you will see that there is a major drop at first, and then it decreases slowly during the infection due to mortality. So this was the parasite dynamic that we cannot observe, but now we'll go to the variables that we can observe. So the blood hematocrit and the fecal egg count. So this is the outputs here. So you see that the blood hematocrit decreases slowly first because the larvae established, they don't have much effect on the blood hematocrit. It's essentially the the adult parasites, the adult female parasites that have an effect. And you see that anemia is increasing, and hematocrit is decreasing. And you see here the fecal egg count. So it's a lot. And usually when we observe data, it's not normally distributed. So we transform the fecal egg count to get a more normal distribution. And unusual transformation is the logarit transformation. So this is the log 10 transformation of this data. And we will use this one for the next steps. So now we have run the parasites model. And I told you that there are two parameters that could be used to simulate variation in host resistance. So there was variation in the parasite establishment and variation in the parasite fecundity. So we'll try to play with these two parameters and see how does it affect the outputs. So we'll do the same as for the beta protein. Okay, so normally you have this figure. Yep. So here you have the number of larvae established, the number of adult females, and here you have the fecal egg count and the blood hematocrit. And you can see that if you play with the larvae establishment, so this is the first parameter, you basically change the number of parasites that will go into the host. And this will have a long-term effect on all the parasites dynamic, including the number of adult females. And you see that there is obviously an effect on the fecal egg count, but you need to take into account that the scale is not a linear scale, it's the logarithm of fecal egg count. And then you have the effect on the blood hematocrit. So you see that if you increase the transition rate, you actually promote the establishment of the parasites in the host. So more parasites means more loss in blood hematocrit. Okay. So now we'll move to the second parameter. That was the host parasite's fecundity. And we assume that there is a host variation in terms of parasite's fecundity. Okay. So this is the outputs. Yeah. So higher value of parasite fecundity have no effect on this variable. Can someone explain why? Well, just try to. So we change this parameter, which is the fecundity. And we observed no effect here. So this is because we only played on the late part of the process, and the larva establishment has not been affected. So this is just a logical consequence. So you can see that you have no effect on the establishment. However, you have effects on the number of adult females, and accordingly, you have effects on the fecal egg count, and also on the blood hematocrit. But the difference compared to the previous scenario is that in the previous scenario, we have only differences in terms of level. And here, we have differences in terms of growth of the parasites. That means that the parasites, they grow more slowly when you affect the fecundity compared to the scenario when you affect only the establishment. So there's two parameters, establishment and fecundity. They control different aspects of the parasite dynamics. And this is something that is very key when you are interested in host resistance, is that the host resistance mechanisms, they are not very well known, and they can affect different parts of the parasite cycle. So it's very complex. Okay, so now we can move to step 2.6, when we will also include the host immune. Sorry, yes?
Can you show me back the
slide?
The slide, yeah, please. So, KF, what was the value of KF before we decrease it?
It was the default value. I don't remember.
Oh, it's my fault. Okay, now it's next one. Okay, no, that's fine. Thanks. Because I didn't understand why all female non-laying was becoming laying, but it just like slow down this curve, and at the end they will all become laying.
Yes, basically we assume that the effect of the host will be to reduce the transition from this compartment to this one. So it's like the host is locking the parasite into a specific stage and avoid to become a prolific adult. So here we have modeled this one, the parasite dynamic, but we have not introduced yet the immunity. So this is the thing that we will do now, the immunity. So we have seen that there are two parameters that can be subject to host resistance. And we will assume that these parameters, they can be controlled by some immune variable. We don't know what it is exactly, but we call it immunity against establishment. And we call this one immunity against fecundity. And it's an interaction. So this means that you have an action of immunity on the parasites, but the immunity is also stimulated by the presence of the parasite. This is why we have an interaction. So basically, the immunity against establishment is stimulated by the number of larvae ingested. And in this compartment, I have a mistake. It's not stimulated by this compartment. It's stimulated by the number of larvae established. So you can correct it on the slide, but it's the number of larvae established that stimulate immunity against fecundity. So then the higher are the values of immunity, the lower are the values of the transition rates. And a third assumption was that this is also a promoter of this response. So it means that if you do not have some level of immunity against establishments, then you cannot simulate immunity against fecundity. So, yeah, I won't go into the details of the equation, but just to know that we have equations behind the interaction. So we have equation for the immune response. So how the immune response grows according to the presence of the parasites. So the parasites here is the number of larvae ingested. So the higher the value is of parasite ingested, and the more you will increase the immunity against establishment. And when you have an immunity against establishment, it will decrease the value of the transition rate from ingested to establishment. And basically, we have the same for the fecundity. And we have parameters that are constant among animals, and we have parameters that will vary among animals. And this will help us to characterize what is a host-resistant animal and what is a host-susceptible animal. So we'll play with these two values now in the script. Yep. So you can jump to the script. So now we'll import our model with the parasites and the immunity. So this is a new model. And now you have the vector of initial conditions. So it's the same as before, except that you have two additional variables, the immunity against establishment and the immunity against fecundity. So we'll change the default values of these two variables to play with. And now we'll try to run the model. And now we can try to visualize the effects. So this is basically the plots that are corresponding to the equations that I've shown you on the previous slide. So what is the immunity and what is the effect on the establishment? What is the immunity against fecundity? What is its effect on the fecundity rate? And you can see that the fecundity against establishment, it increases at the beginning, and the effect is at the beginning. And the immunity against fecundity, it increases at the late stage of the parasite infection, and the effect is later. So the difference with before is that we have introduced a dynamic immune response. Instead of considering constant parameters, we have considered dynamic change in immunity that help us to draw more complex parasite-host interactions. Yep. Is it still?
The two graphs, do they look opposite each other?
Yeah, but well, actually, it's more or less this because the higher the immune response, the higher will be the effect on the parasites, and the effect is just a reduction in some transition rate. So yeah, it's almost the opposite. Yep. Okay, so well, now for later, if you want, you can explore the different effects of varying the values of the immunity parameters. So to do this, you will use the same function that I showed you before, the Laplace function with different value of parameters, and you will simulate this for different values, parameter values, and you will see what are the effects. Well, we won't go up to this stage. We'll go directly to the last stage. So the last stage will be to integrate the host-parasite model with the energy balance model. Yep. So far we have done this step. So the step 1 and 2. But still there is something that is missing in this model, if you have followed. This arrow, which is the energy that is going from intake to immunity. So this is the final step. And yep, the key question in this study was to draw some conclusion about the magnitude of this cost. So basically, this is equivalent to consider our energy balance equation and to add this component, the energy allocated to immunity. And we'll consider that this will be just the sum of the immunity against establishment and immunity against fecundity times an energy cost. Just like to deposit 1 kilogram of protein, you need some energy. You could also consider that to develop some immunity, you need also some amount of energy. So this is the last stage, so step 3. And in this last step, we will simulate the complete model, and we'll try to then simulate the consequences of different parameter values, and then finally estimate all the parameters together. Yeah, so step 3. So this is the complete model. So basically, it's all the equations of the previous ones that have been compiled together. And then you can define your vector of initial conditions. So it's the same as before, except that we also introduce the initial state of the energy balance, so protein, wool, and lipid. And now we will run this model. Yeah. And now we are ready to observe the effects. So for fecal count, blood hematocrit, OK. So now we have our 4 outputs: fecal count, blood hematocrit, body weight, and back fat thickness. And you can see that the fit is still not so good. So we can try to find better parameter values. And there are 3 parameters that we will try to modify. The first will be, the 2 first will be the immune parameters. So the immunity against establishment and the immunity against fecundity. And the third will be the protein growth rate, as we did before. So instead of doing just this one, we'll also consider variation in immunity. So this is an additional consideration compared to the initial scenario. So 3 parameters to fit. We can calculate the residual sum of square for these default values. Yeah. So you can notice that it's higher than before, which is normal because we have 2 additional variables. We have fecal count and blood hematocrit, whereas before we only had body weight and back fat thickness. So this is just normal. But still, we can see if we can improve the fit. So we'll use the same algorithm than before. So this, it will take a little bit more time because I need to explore the parameter space while it's done. And now you have the previous residual sum of square, the new residual sum of square. So there was an improvement. And now we need to check if this improvement makes sense. So we'll visualize the model with the new parameter values. OK. So basically, what we do here is that we replace the 3 parameter values we were interested with the new values. Yeah. Ah, OK. Thanks. OK, so we have the 3 new parameters. And now we can calculate the model. Okay. So this is the new one. Where is the new one? Here. Okay. So yes, I forgot to put the previous simulations, but actually you can see that the fit is better, especially for the parasite variables. So the fecal egg count and the blood hematocrit. And you can see that still the body weight is not very well predicted. But this is just something that we have to accept in this model, unless we try to explore other parameter values. But sometimes it happens like this. Well, we have some parameter values that we found, for instance, in the literature, that present some variation between individuals, and we did not consider variation here. So this might explain why we have a body weight prediction that is different compared to the observation. For instance, we have a variation in feed efficiency that was not accounted in this process, and which could explain the deviation. But well, you could see here that we have a model that is able to predict the response for a particular sheep. But now we can see with a different sheep if it works also. So this sheep was a susceptible sheep because you see that there is an infection that is going on. And yes, just before going to another sheep, we'll calculate what is the energy allocated to immunity, because this was a main question in this model. So the energy going to immunity, this equation, it's basically this equation here, energy allocated to immunity. So I calculated. And I also calculated the energy intake. And now we can calculate the energy allocation to immunity. So you can see that this is the amount of energy allocated to immunity for this particular host. So it's in megajoule. And this is the same graph, but in proportion to the intake. And you can see that actually it's a small proportion of intake. It goes up to 3.5%, so it's very small compared to the growth rate or to the maintenance. But this was a susceptible host, and this is maybe why this value was so low. So now we can try to do the same thing, but for a host that was resistant. So this is why I also proposed you data with for a resistant host. So it's another data set that you have in your files. So if you upload this file, we can do exactly the same that we did before. So calculate the splines of the feed intake, set the parameter values, define the vector of initial condition, and then try to estimate the 3 parameters of interest. So we try to do this. Okay. And now we have a fit. So we define our new parameter values, the 3, and now we run our model with this new parameter values for the resistant host. And this is, yeah, so these are the results. So you can see here that the fecal egg count was almost 0 during all the infection. So this was a very resistant host. And that the blood hematocrit was not even decreasing. It was stable or even increasing. But, well, for blood hematocrit, you also have some variation that are not due to parasites or, well, there is random variation. And you can see still that the body weight is not well predicted for this animal, and the body fat thickness is more accurately predicted. So with this model fit, we can try to estimate what is the energy allocated to immunity, just like we did before. So let's calculate in the same way as before. We have the energy intake. Up, duck. Yep. And now you can notice that the energy allocated to immunity is higher. And in terms of percentage of the energy intake, remember before the maximum was 3.5%, and here it's more than 8%. So it was twice as much as before. Still, it's not very important, but it's not negligible. Okay, so this was the last step of the script. So the objective was to give you an overview of the modeling process and try to fit with different data sets. So you have all the scripts. You can do the procedure again, try to play with the parameters. Feel free to ask questions on this later on. I will be available. And if you have questions now, yep, I will be happy to answer. On the code or more general questions, I mean.
Do you take into account the fact that resistance might allow better resource allocation and absorption of nutrients?
Yeah. Actually, yes, the resistant costs are very complex because it can be, for instance, not only energy, it can be protein. So there are different nutrients that can be concerned. And you have also different effects on different parts of the nutrient utilization. And for instance, we know that the parasites will have an effect on the energy absorption at the level of the gastro-intestinal tract, and this was not accounted for. So this will, for instance, affect the energy efficiency of the conversion. Yep. Another effect that was not accounted for is that when you get infected, your intake usually decreases. So this is why we use directly intake as an input, because we didn't want to make assumption on the intake depression. Yep. But there are a lot of assumptions that we could do regarding the infection process. It's very complex.
I would like to know about repeated infections. If this changes the model, if you get infected on day 1 and then again on day 5 or 6, it will also change the life cycle of the parasite. The females will mature later on.
So the day of the infection here was defined relative to the larva intake. But you could have different infection. Well, in natural conditions, you have repeated infection. Every day the sheep encounters parasites on pasture. So you have different infection times. This was not accounted either. It was a single infection. But when you have a repeated infection, the main effect that you will observe is that the immunity of the host will increase progressively, and this will progressively make the host more and more resistant. So this is why the lambs are the most susceptible after weaning, and then progressively they acquire their immunity, and parasitism is not a big issue except at some stage. But yeah. Yeah, but still the problem is that we do not have a good biomarker of immunity. With these parasites, it's, yeah.
Do you know if there is an immune memory for this parasite?
Yes, it's exactly the acquired immunity. It increases, and then there is a memory. When the host gets reinfected, it will react faster than in a naive host, for instance.
I'm sorry, but I would like to make a comment about the natural and the artificial infestations in terms of what animal breeding means. Because artificial infestation has lots of value for experiments in terms of it's part of the protocol that helps to standardize your conditions so you know exactly the burden, the work burden that is going to affect the animals and how to do that. The natural infestation is used when, because I'm part of one of the classes that is there, so from the thing is that when you collect information that is recorded directly by breeders on their own lambs, is the only way to do it is based on natural infestation and trying to have this protocol of ensuring that at least you have certain level of infestation in the pasture. So we record the data knowing that they record, because the breeders record the data based on certain levels of infestation in the pasture. Is far away from knowing exactly the level of infestation that is an artificial infestation. But given the structure of the breeding programs, it would be impossible to have data recording in thousands of animals, in hundreds of breeders or farms, based on artificial infestation. So the best way is to build protocols that ensures that that FEC is recorded when you have at least certain level of infestation in the pasture, because it wouldn't be impossible to be distributing homuncus around the country to be sure that all the breeders would be delivering FEC based on artificial infestation. But it's based as an adaptation. It's the best protocol, but the big difference is on the structure of the breeding program. So it's impossible to generalize which from a breeding point of view could be the best way to record the data, because it's number versus also accuracy or possibility of data recording versus central progeny testing like you have here in France. So there's advantages and disadvantages from both points of view. It's clear that for certain experiments like modeling, the clear way is to use artificial infestation. Based on the data that you presented, I wanted, because you always mention the percentage, the energy in the diet, the energy that the animal is using. For parasites, the percentage of protein in the diet also has an impact on the immunity or the response. Have you analyzed that in terms of the diet?
No.
Because it's also one of the practical ways to control.
Most models actually focus on protein, because in some country, protein may be quite limiting on pasture. In UK, I think, Scotland, yeah. But maybe it's less a problem in France. And in these experiments, so we focused on energy, because the diet was 100% concentrate. That was very dense in terms of protein, and energy was limiting. And there is some evidence that energy is also important for the parasites. But it's true that we would need further experiments that that well, try different values of energy and protein ratio. And well, it's also important because the parasites' impacts on the long term have not been shown, but presumably you have an impact on the protein growth, and when you have a lot of experimental data that shows that growth is affected on the long term. Here in our case, the sheep, they were even beginning fatter during the experiment, even if they were parasitized, so it was not very harmful for them. And regarding the breeding, the breeding strategy, this has been shown to be effective on the field. So
I'm not questioning the design of the breeding program in France. I just explaining that it's impossible to have artificial insemination as a way to record FEC in breeding programs when the breeders are recording the data on their own lambs, and you're talking about thousands of animals per year that are being measured, and at least hundreds of, maybe in Australia even more, breeders working across the country. It's impossible. It's not practical to have that implemented.
Yeah, in this case, it's only one center that.
That it makes that possible to use artificial infestation, what is completely impossible to be used in other situations.
But Ellie, if you take a genomic view of this, and you can do something like this on a smaller population, so you can identify which parts of the genome, and you then use that in the broader population, assuming you had a significant genome for.
Well, if we have the possibility of doing, we are actually building, we are having genomic selection for, at least in some of the merino breeds, with the possibility of including FEC now, and the training population is built on FEC collected on natural infestation. At the same time, you are focused here on amoncus. And actually what we have in Uruguay, and the protocol tries to record at least is one FEC or two measurements of FEC. Selection for FEC works because there are selection lines that have been selected for many years, for generations for that. We tested the feed efficiency of those animals, and there were no difference in the feed efficiency. We expected to see difference, but we didn't see difference between resistant and susceptible animals, and it's something that, it's like we still have a question mark open there in terms of saying, well, actually what is really going on within the animals that we did. No, we didn't measure anything, but we did all the measurements in terms of animal composition, body composition, with the back fat, with the muscle area, feed intake, growth, everything, with artificial infestation in that experiment, and we didn't see difference. So it's like in some ways, good news, because we don't have trade-off, or we haven't identified trade-off. But the question mark in terms of what happens in terms of the balance of what is the cost of the immunity, or how the internally or metabolically that works, is still open, because we didn't got clear clues, because the behavior science.
When you do your FEC, the collection from the field, do you differentiate between the different parasites, or you just have, because you can have strongylids and like trach, which you are talking about, which will have something different from Haemoccus. One Haemoccus can shed about 10,000 eggs, so it's a different parasite. You would need them to plug those in your breeding program first to see whether you've got different irritabilities with specific parasites, or if you combine all the eggs, what is the difference?
No, for the normal recording for the breeding program, for the genetic evaluation, there's no identification of the species. It's just samples and counting eggs. But for all the experiments or sometimes monitoring in some specific farms where other things are being collected, then they work on the identification of the species. So that's why we know in the different regions more or less the percentage, and also the variation between seasons or between the different dates in terms of the percentage of the parasites, of the different parasites, but not for the normal recording for breeding purposes. It's impossible to have. The lab wouldn't be able to do.
No, but you have, they group them. I was working with the British breeding program at the beginning of my career, and even in Zimbabwe, like I was telling you that we used to measure them. There are certain groups you can just group as strongylids, because they've got very little eggs or small eggs, and you can't differentiate, but there are bigger ones and some that you can differentiate. Therefore, the big ones, they are easy. The small ones, you can just put a number, or greater than so much if
no, it's not. It haven't been differentiated so far.
Because each one will be different, depending on the region. Like you're saying, the rain season and things like that, you have different things. If you don't know, I think you're just lucky that it probably is very similar across all the regions. So you're pulling everything together.
The big effort now, that's something that I didn't mention the other days, in terms of developing genomic selection for all these traits, no? For feed efficiency, but also for genetic selection for resistance to parasites is implementing that, and is basically merino. The first genomic EBVs for merino breed are going to be published this year. So, but it's still the thing of knowing what is happening in terms of putting together the information. Sorry, I didn't want to.
Yep. Any other question before we finish this day? Okay. Well, thank you for your attention.